Good afternoon, everybody. So this project is still in its infancy and I don't have any results, but essentially what I want to try and do is highlight some of the practical applications for the industry by sequencing the genome and the transcriptome of false codling moth. So I'm not going to go into detail about the beast. A lot has been said about it by both Dalian and Marlies. Essentially, I just want to highlight one important thing, and that is that FCM has developed insecticide resistance, and this has turned the focus to alternate controls. And despite this, we really need more firing power to combat FCM, especially in the light of exports for this specific pest. So just very briefly, essentially this whole project hinges on what we refer to as the central dogma, and this is the basis of genetics. And essentially it is this flow of information from DNA all the way through to proteins, which are the end products of the gene expression. And these proteins is what forms the basis of all biological processes in organisms. So essentially we are interested in what are the proteins that are being expressed at different endpoints. So in this project we are interested in a branch of molecular biology called the omics, which sounds a little bit like a band name, but essentially we're interested in two of these. There are quite a number of them. The first one is genomics, which is essentially concerned with the structure, the function, the evolution, and the mapping of the genome. So just what is the blueprint? And the second is transcriptomics. And in the transcriptome, we are interested in the expression of the genes. I've mentioned before that genes are differentially expressed, which means that not all genes are expressed at all times. So essentially the overall aim of this project is really just to develop a toolkit or a toolbox to support novel control methods, but to also better understand the rapid evolution of pesticide resistance, as well as environmental stress adaptation. So just methods really briefly. Essentially, we'll be extracting DNA from false codling moth. This will be sent off to a genomics company called Macrogen, and what Macrogen will do essentially is they will use the DNA that we've extracted in our laboratory, they will fragment it, they will sequence it, and we will get these reads back. We will align the reads to each other to make essentially a long string of DNA repeats, and we will then assemble this genome, and then we'll end up with the entire blueprint of the false codling moth genome. And this allows us to use this blueprint in many downstream applications. One of these downstream applications is the sequencing of the transcriptome. And specifically, one potential experimental project that we are going to be tackling is looking at the interactions of insecticides and temperature. As I've mentioned before, FCM has developed insecticide resistance, and what we are able to do with sequencing the transcriptome is we are able to investigate some of these genotype environment interactions. So the environment interactions we are interested in here is the insecticides and the temperatures. So we'll have high temperatures, low temperatures, with and without insecticides, and we will then extract the RNA, and the RNA is where the information lies of what genes are being expressed. What we are able to do with this information is essentially identify differentially expressed transcripts and then also draw these expression patterns. And what this will show us is, for example, if FCM is put into an environment where insecticides have been sprayed, then we are able to see what genes are being upregulated and what genes are not, for both those that have insecticide resistance and those that do not. So we'll be able to see what are the genes that are on and what are the genes that are not on. So the question then is, what can we do with this data? So not only will it help us to really understand how FCM survive and adapt to a suite of pesticides and also environmental conditions, it will also allow us to develop novel control methods, which I'm sure most of you are really interested in. There are a couple of these that I've listed here, but there are many others. So the first is RNA interference, or RNAi for short. Essentially, RNAi is a gene silencing. So if you know what the gene is that is responsible for insecticide resistance, you can actually knock down that gene, which means that the active ingredient will work again. The second is CIT, and the last one that you guys may have heard of before is called CRISPR. It's clustered, regularly interspersed short palindromic repeats. Essentially what CRISPR does is, in the figure that I show here, is you can use the desirable elements in your transgenic organisms that you can introduce into the wild population by using a very small number of individuals. And this then allows you to introduce the specific element throughout the whole population. What this allows you to do, for example, there's been many trials in fruit flies where they are introducing skewed sex ratios, so basically only males or only females or high proportions of each. And then the last one is basically where they are making some of the genotypes completely infertile without, so essentially it's just inherited from generation to generation. And that is it. Thank you.